So I hope everyone has relaxed enough, hopefully. Now to the second part, which will be about image analysis this time. So let's begin. So this morning, as a quick reminder, we had microscopy by Soltan, and now we are handling image analysis, and this will not yet involve anything JIPipe. This will be the basics of image analysis. So you learn the general terminology. So you learn about the general techniques that are the same between a lot of image analysis tools. And then later on, tomorrow, then there will be actually the course where you actually use JIPipe. As a note, you see that this box with these keychains, feel free to take the keychain. This is official exclusive JIPipe merchandise that is for this year, specifically made for this course. So this will not return this at least this design. So let's start with when you get the images out of the microscope. Then I also did it myself, although I'm a bioinformatician, is you analyze them often enough manually. You can just look at it and then count it manually. You just, for example, with these bacterial colonies, and you you use a pen to count the colonies and so on. But you quickly hit a limit with this. For example, I have here a confrontation assay that has 4 channels, red, green, blue, and gray, where you have different elements or different cells being labeled. Red macrophages, green are the fungal spores, and blue are the fungal spores that are also outside of the macrophages, and gray is transmitted light. And the task would be, at least in this specific data set that is published here, was to count which spores are inside and inside the macrophages, and keep track of the color, because we need really to know the color to really for 100% sure know if it is inside or outside. And you can absolutely count this manually. But you quickly, when you do this manually, run into an issue where this is maybe not so straightforward as you think, not only super time consuming, but there are sometimes decisions that are a bit researcher specific. So for example, I have here some image examples cut out from this specific data set where it's a bit confusing, and then you ask 10 people and you get 15 answers. For example, how many spores are those? It's directly at this tiling border. Do you count it, do you exclude them, but then you skew the statistics maybe if you exclude them, you never know. Or if you find structures that are a bit ambiguous, if they are really inside or outside, or is this even a spore? Because it can be also a question, is this like an artifact that was left over, or something, the coloring labeled something that is not actually a spore? And then here again, you have cases, you can really badly see it only on the screen, is it really inside or outside? And as mentioned, is that you will disagree with other researchers about this. So you ask your colleague, then they will probably produce another number. Or if you ask yourself, maybe next day, or if you have a good day or a bad day versus the last time you have counted it, then you might also produce a different number. Or if your screen is a bit different set up, so the colors are a bit washed out in the screen settings, then you might also count it differently. And this is quite inconsistent. And then we have the big issue, as in really big, because what I showed you is of course not one image. It's just a cutout of one whole image. It looks like that. This is a real screenshot from one of these visualizations produced from this data. But then you not only have one, you have like a folder with hundreds of them. So you actually need them to collect enough information about your different conditions so you can make good statistics on the data and then have fun counting these. So this specific colleague, from what I've heard, he sat there like for 4 months and counted them manually, which is quite time consuming, you can imagine. But this is why we have automated methods that probably help you with that. So in manual analysis, you are struggling a bit with researcher bias and variability in general. You make decisions, but then on what do you make the decisions? You have hidden factors that are dependent on different conditions, on your eyesight, on your screen, on your mood. But then you have as alternative tools that do it sometimes. You need to give it an additional information manually or fully automated analysis. So computers would then do it for you, and they just do raw processing for you. They have generally no variability. There are some exceptions as well, but let's exclude them for now. Then the computer algorithm doesn't mean it is perfect. It can make issues or wrong decisions or wrong segmentations, but then they can go in there and then actually identify what it is and then correct these issues. And the hidden factors in your head are replaced by, for example, program code that we can actually look at, and then we can really identify clearly what criteria are used, except if you have like something that is machine learned. It's a bit more complicated, but for classical algorithms, this is still the general mode. And computers, don't underestimate it, are super fast as well. What takes 4 months to count manually, a computer takes maybe half an hour, and then you get a good result out of it if your algorithm is designed well to account for all the differences in the images and it's well calibrated. You can then use all kinds of different image analysis tools that you can pay a lot of money for. These prices are actually not one-time prices. These are usually yearly prices as well. For example, Imaris, that we have here in our department, MATLAB we have also, and Huygens we have also for deconvolution. Velocity, I personally don't know, but these are realistic price ranges of what you expect from these commercial tools. And they do it for a reason, because they are very, very good and have a lot of integrated automatic functions to get you to your result and can handle large data as well. But this is really expensive. You cannot just download it at home and then try it out. So there are also a lot of open source tools that you can use to do the same tasks, or sometimes a bit more, sometimes a bit less. They all differ a bit. But the general thing is they are for free and you can just download them. Very commonly used is Cell Profiler, very, very popular. IC is also popular in some specific communities. NIME have their own image processing, although they are mostly focusing on data processing in general. So working with statistics and tables. And then you have the interactive tool that is very popular, ImageJ or Figge, the distribution of ImageJ with lots of plugins. And then built on that ecosystem also JIPipe, which is this course, this course is about that one. So, but before we use any tools, we have to first look at what is actually an image to understand really what these tools do. And so we can really leverage the usage out of them. And this includes how is actually an image represented in computers, what are dynamic ranges and bit depth, how do they fit together, which file formats you should use, for image processing, what are the actual basic steps that you can always use as scaffold to get your image analysis going? How do you actually enhance images so you see the features that you are interested in, thresholding or segmenting the parts out of that image that you are interested in, and then processing steps that you may or may not need after thresholding to correct mistakes, for example, or correctly find your objects. So what is an image? And if you zoom in very much in any image editor, you see that an image is just a grid of pixels. Looks like that. If you zoom very much in, each pixel in general has a value. And this value is not infinite. It is not possible to put something infinite into a finite space like your 16 or 8 gigabyte of RAM that you have. And the actual limit is defined by something that is called the bit depth or the data type. And they are very closely aligned concepts to each other, bit depth and data type. I will explain them in the next slides. So, and how the computer save it so it's really fast and can actually work with images fast enough is that each pixel in an image has the same fixed data type and the same fixed bit depth. And then what you will find in general with many image analysis tools, you find, for example, that the values can be integral. So they can be whole numbers, 0 or 1, 0 to 256. Then there are images that can store real numbers, so with comma values as well. And then also very common images that are storing colors that you see on your screen. These are colored images. And then here in this example, if you take this pixel out here, this has the value 10. And then a bright pixel, a high value like 201 in this 8-bit integer image. So what is then this bit depth? And again, we are working with finite memory. You have to basically assign to each pixel how much space in memory it will take up. And they do this because this is super fast for computers. When there's something that is evenly spaced, basically, then the computer can pre-calculate where to access information very fast. This is not the case when this is not given. The bit depth is basically where you say how many bits, so the most basic unit a computer can work on, 0 or 1, is assigned to one pixel. You will see examples in the next slides. So for example, if I have an image that has a 1-bit depth, then it has basically a storage container where we have only one singular bit, and this one can be 0 or 1. And then you cannot store any other numbers. It can store only 2 states, 0 or 1. If you have an image where each pixel would hold 2-bit, so would be a size of 2-bit, there you can hold up to 4 values. So like with this binary notation, there you get already from 0 to 3. And then you can extend on it 4-bit, you get from 0 to 15, so 16 values. And then 8-bit, this is usually the lowest image data type that you work with, 0 to 255 in general. And you see it everywhere. An image is prominently called 8-bit image. It's this one that has 8 bits assigned for a pixel, and it can store 0 to 255 as values, and not anything more. But this is not so simple either. You can also say, and the computer will do this for you if you set this up correctly. It's not of your concern, it's concern of the developer that develops the image analysis tool. They can say, if you have 8-bit space in this pixel, I can say, split this up virtually into 2 4-bits. In this way, we can store 2 numbers within these 8 bits. There's one way to basically, we have a different interpretation of these pure storage in bits that is available. And then I have a bit organized and a bit weird. Then we have also 32-bit images, and they are a bit interesting as well. So we have 32 bits of storage, but there are theoretically also not that much. Theoretically, you can go from minus infinite to infinite in the allowed value range, but they have some constraints where they basically get less precise the further away you're from zero. You have an explanation, and then how this works inside the computer, the so-called floating point numbers, is they assign the bits to different roles. One bit says it's the sign of the number, it's a negative or positive number. Then they use an exponential representation, where then they use a few bits to say this is the exponent of the number, and then the fraction, and then this way it can calculate a real number with comma values that is more or less theoretically negative to positive infinity, although less precise the further away from zero. And at some point you break, basically the structure breaks because you have no space to store this image anymore. And then you get weird results that come out. So better don't reach the limits of these floating point numbers, but it's very unlikely that you will reach this. So to quickly summarize, because this is a bit of a weird concept of thinking differently about images and how they are stored. BigDev in generally is how much memory do I have available for each single pixel, and then the data type, so let's call it like that, is basically how do we interpret it. Like in this slides before, we can say this is either one number from that one integral number from 0 to 255, or we can say these are actually two numbers stored in the same space, reinterpreted, but then of course we are size limited, we can only store from 0 to 15 in each of these numbers. And you can actually also make an 8-bit floating point number as well. This is actually common in AI now. They found some way to basically squeeze a number that has a real number with comma values into 8-bit somehow. And it works, because the AI stuff works quite well. So when you're working with ImageJ, especially the common BigDevs and data types you will encounter, there are only a few, because ImageJ has very limited support for them. So the most common one, 8-bit images, they just call this in ImageJ directly 8-bit, but it means actually 8-bit per pixel, and we say it's an integral number. And there we can store values from 0 to 255. Then the 16-bit integer images, you can also switch it in ImageJ. They can store a larger value range as well, and this is very important when you have data that you have that is really low contrast. There you can just have more space to put values in there, and then so you can really also capture quite low contrast structures that would just not fit in 0 to 255. For colored images, ImageJ says in ImageJ they call it 24 bits, which is theoretically 3 times 8 bits. 3 times 8 is 24. And there you would, they say each of these 8 bits, so each of these 24 8-bit chunks are set to be RGB channel. I anyway will show you the RGB color space in further slide. And then 32-bit in ImageJ, we have this floating point numbers. Theoretically goes from 0 minus negative infinity to infinity, but you will hit a limit at some point with those. And this is basically what happens in ImageJ. So then colors. There are actually different color spaces around. The most common one is RGB. This is the one if you that is basically used everywhere. If you go and take a magnifying glass and zoom on your screen, you see this pattern, red, green, blue, or some variations of it. This is basically how everything represents colors. But for scientific applications, there are also other color spaces as well. that I will explain in further slide. For RGB colors, as mentioned before, you can say these are together 24 bits they take, and then each of these and the whole 24 bits are split up into 3 times 8 bit integers, one for the red, one for the green, one for the blue channel. As a side note, in reality, they never do this because there is no way for a computer to easily have something that is 24-bit in size, so they usually add one 8-bit additional to it. Then you have 32-bit, and this is just some implementation detail that then the computer has usually one additional channel. For colored images, they may or may not use it. For images that have transparency, if you basically take additional 8 bits and add it on top, they use this for the alpha channel, so for the transparency channel. But ImageJ cannot do this, it cannot handle transparency, so it just ignores this additional space that it usually just allocates for itself. It makes no sense to just limit yourself to 24 because there's custom hardware causes. It is faster to work with 32-bit than with 24-bit in computer hardware. So, then regarding colors.
I have some questions.
Yes.
So generally when I took some image, I went or with the 12-bit or with the 16-bit, and then what does it mean? They were taken with 3 channels as well, so blue, green, red. So there is a different allocation, I don't know, 12, it's
yeah,
there's integer, but it's a different one.
So there will be something else. This comes a bit later,
okay.
And this is a super confusing concept because this is also called channel and something different from that. This is super confusing, but I will explain it in when we go to file formats how they actually store it. This is mostly not a concern. So when you have a 12-bit image that has 3 channels, the channels are stored in a different way that it does not affect your range that you have. But this will come later. So first I will explain a bit false colors. So when you look at the image on screen, in an image viewer, usually it will apply some kind of false coloring that looks nice often. But in reality, the pixel values are still grayscale. For example, here this is like an 8-bit integer grayscale image, but the viewer, I told it to use a so-called lookup table or LUT to make it fancy. This was actually specifically for some media. They love this when this is super colorful. So there was some publication like for TV or something, or for the press something, and it makes no sense that it is colored here like that, but it looks nice. So don't underestimate that. Also, sometimes it helps you see things a bit better because our human contrast system is really evolved to handle specific colors better than others. So, and regarding false colors, there you can apply different lookup tables that may or may not be useful depending on your use case. There are some ones that I took here from some website. And generally what they do is they assign to a grayscale value. Generally they limit themselves to 0 to 255 for performance reasons, to a RGB value, and then they display it screen only. If you have a 16-bit image, depending on the tool that shows it, they may basically internally convert it first to 8-bit. So this lookup is fast because you can imagine looking through a table of 255 values is way easier than a table of 65,000 something values, and it's also smaller. And you don't see it anyway because our computer screens anyway have only 8-bit RGB, so you just waste a lot of compute and space. So this is generally how all lookup tables work in all tools. It really looks like that, and you can even make custom ones in some tools. So, and then we are back to the image in general. So when you're working with your microscope, it will output an image. And this we come slowly back to your question again with these data types and bit depths and so on, how they relate to your microscope in the end. It will output images according to your settings and save it out. And then sometimes it's just, if you are not aware of it, you cannot break physical laws of information theory and so on. Then you save it from the microscope in a low bit depth, like 8-bit. You just increasing the bit depth later on in Photoshop or whatever tool, it will not recover any information. It's gone. You can reduce information always, but you cannot just invent information back. You cannot go back in time and get the information. So you have to balance always the bit depth, its size requirements. So the higher, the more memory you need, versus how much information you actually need to represent your data. For example, then you have like these uncommon ones like 12-bit that image cannot natively handle, and then just upscales it to 16-bit. But then you have to be careful regarding the file formats that you work with, because not all of them can handle all bit depths and all data types internally, and sometimes they lose information that then is just gone when you saved it out wrong. And then when you go then to the image analysis and you suddenly miss this information, and it's gone. You cannot recover it. You might have to repeat it, your experiment. And there are many, many different file formats that you encounter everywhere. It's like TIFF and OME-TIFF and PNG and so on. The wrong one is meant for you will lose information, and then when it's gone, it's gone. And unless you have a backup of whatever file that has more information. And that's why I have made this small table here where you can quickly glance at what purpose you want to use a file format and then what file formats basically fit. So for anything web publishing suitable for website, there you can use GIF, although GIF is quite ancient format and I wouldn't use it anymore, or JPEG with its blocky compression artifacts. Publications, bitmap, while rare, still technically possible, or PNG, the common standard now, because PNG can lossless compress as well. Then content creation, you have these Photoshop documents or the from the GIMP image editor, its own file format, or if you're making figures that have vector art, you can use SVG. For example, Inkscape makes these for posters. But then when you're actually doing science and still processing the data and not yet anything that should be published or go on a website, then if nothing else is available, then just use TIFF, because TIFF can hold actually a lot of information thanks to its metadata system. And I will explain in the next slides how it actually stores images. Then there's a one step up from TIFF. This is OME-TIFF standard that is made for, it's basically an extension of TIFF designed really for microscopy work, where it can store actually a lot of metadata that your microscope produces, and the people who develop this format actually work together with the microscopy manufacturers that the data is actually stored in there and standardized and properly organized. Or the new format, OME-SAR, which you probably have not encountered yet because it's not there yet. I will explain it briefly. And then ideally, when you are archiving something anywhere, better have always the native format. So even if you use Omero, like an image database that is remote, put the native format there that the microscope produces, the lib files or the CZI files, because they store every metadata. And then you can later go back, you can later down convert it to something that is maybe more compatible, because these native formats only few softwares can actually handle. For example, ImageJ can convert them internally into its own format, or Imaris can do the same thing, where it's like converting into an IMS file. But keep them around because then you might need to go back and extract some information that is very specific to your case, and then you regret it if you deleted them. And then the others you can use for intermediate processing.
I have another question, but I don't remember. EMG and BMP are vectorial as if or not?
PNG and bitmaps are basically formats that are more for publishing, so therefore for web, so it's literally in the name, portable network graphics. What PNG does, so it cannot, for example, handle 3D images. It will just flatten, it usually takes the first slice and then, or it was just ever out for you.
So I'm losing in any case information.
Yeah, you lose a lot of information when you use them. They are good if you want a figure in the end, these formats. Although bitmap are rare now because they don't have all features that PNG has. PNG has transparency as a feature. So, and TIFF is like also very common. So, for example, for printing, they like to use TIFF as well. But this can actually hold way more than just use for pure printing. Or to have. Did I answer correctly what you?
I don't know. But okay.
Because I have some ear issues.
Okay.
Yeah, I have difficulty hearing people in this hall, in this room here. So it's generally, so this comes in the end out when you're making your publication, your journal publication or your poster or whatever. These out. But this one you can actually use for real scientific stuff. And you will see now in the next one why. Because you're mostly working with multi-channel or multi-dimensional images all the time, which is now the standard. And it's actually not so trivial to store them in a way that the computer can work with properly. So there are actually tools that can basically store them as you think. So you store a three-dimensional image as a cube, basically, or a hypercube if you four-dimensional images. But the most common way how they store multi-dimensional images is you have 2D image planes and they basically stack on top of each other. This is still the most common way. You can also split it up nicely and it's very compatible to everything. Sometimes in one folder you have multiple files. Sometimes you have one file that has them organized in some way internally. And this is basically what TIFF does. TIFF cannot only be used for 2D images. It's a very, very broad standard that supports a lot of things and also metadata information. And TIFF can basically store an image stack as well in form of these planes. And then this way you have a three-dimensional image. And the very common standard way of storing TIFFs that have multiple dimensions is, okay, according to, you have these constraints here, but then you say you have three additional different dimensions. And this is where the confusion comes from with channel. You have the channel dimension, the depth dimension, and the frame dimension inside the TIFF. And this is super, super confusing. So first, how TIFF it stores it actually, so it assigns a metadata to each plane that says the channel, depth, and frame location of this plane. So I've written it here out, how it looks like for a standard three-dimensional image. And then some constraints, you don't allow anything that is duplicate, you don't allow gaps, so you cannot just remove this one here and then it's gone. This has something to do how this format internally organizes that in an efficient way that it's fast and works reliable. And then we come to the thing, channel versus channel. And this is the ultra annoying thing that they named it channel. So we have RGB colors, which, as explained, you can represent as 3 times 8-bit integers, and then we have a red, green, and blue channel inside the pixel. But this is something different from these channel locations. This is a location of this whole slice plane. And you can have, according to the TIFF standard, an image that is RGB colors. So it's each pixel stores 3 channels, but then have a multi-channel RGB channel image which has multiple channels. Both are called channel, and TIFF supports it like that, where these are distinctly handled. But you have tools that cannot handle it properly, and then they do something with it. For example, if you use ImageJ, it can handle it properly. You will be able to have a 5-dimensional image where you have X, Y, Z, X, Y, Z, Z, T. That's the ImageJ official order of channels of these slices here, and then and each one can hold an RGB image, which has then multiple. Each one has 3 channels per pixel. But then when you load it in Python, so with SciPy and NumPy, this cannot actually handle that. And then it will basically take these channels here, and then because it doesn't want to lose information, it expands them basically into an extra axis. And this can cause some weird confusions, because then when you convert between Python and ImageJ, this causes some weirdnesses sometimes, that your image suddenly has 3 additional dimensions out of nowhere, and which an ImageJ cannot handle, because ImageJ can only handle 5-dimensional images, not anything more. So it's really careful. And then when you have a 12-bit image that has 3 channels, this is very likely that you have per pixel 12 bits integer, so just one number, a value, but then you have 3 different channel locations basically. So we have channel 1 slice, channel 2 slice, channel 3 slice in there, and then whatever set stack. So in a simple 2D plus channel image with 12-bit, it would look exactly like that. You have then each one is in 12-bit, 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 but then organized like that. So the image tool can access it like that.
Excuse me. I didn't really get what is channel for TIFF.
So TIFF channels are basically just, and this is what is confusing, is just a axis, a dimension axis. It's just the metadata. So they assign, they just call this one channel. So you have 2D planes that are stored inside TIFF, just 2D images. And these 2D images are assigned a location. And the 2 of these locations you can easily grasp. So, okay, the frame, for example, the time location, or the Z layer, when you go up and down. And then they have another type of location, and they named it bit confusingly also channel, which is different from the channel that is per pixel. So this channel location of this 2D slice, it's something different from these channels, yeah. It's just you have to handle it. And the weirdnesses of these different tools that may or may not handle this properly for you. It's just the confusion. But in general, with scientific processing, you anyway don't work with RGB images in general, hopefully, because you have only 8 bits per RGB channel, so you lose a lot of information doing that. You generally work with images set where each slice has like 16-bit, so each one of these slices is a 16-bit image or a 12-bit image, and then they are organized in this structure that is called a channel. And then in ImageJ you can scroll through it with this channel slider, but it actually scrolls through these different separate slices instead of filtering out something from the pixels. It's just the reality of that, how they organized it. And then as a heads up, you will encounter this at some point. That's why I mentioned it, is this OMISA format. Because it's working on the people who are behind it. It's the same people who are behind OMI-TIFF, so the standardized TIFF format for microscopy. The OMI-TIFF format is designed for 5-dimensional images only, and when you have some additional dimensions, you run into problems with it. And this is why the people designed this OMI-SAR format. Is basically a replacement for it. Also, it can not only store 2D planes, it can store the images in a different, more optimal format. And you will at some point encounter this OME-SAR format or OME-NGFF format. You can read up about it and how fancy and great it is, but you will just at some point encounter it because the people who are behind it are working together with the microscopy providers and with the image analysis tools, so the commercial ones, to actually support that. And the great thing that they will have there is not only more dimensions, but also you can. It's designed for remote storage, so you can put it on a server and then access only specific information that you want, or you can also have a multi-scale image where you can, where you don't need maybe the full field of view, full scale of the image in its full large size, but maybe just an overview, and then the format basically natively supports that access to it. I don't have any specific examples about that one, also not in JIPipe, starting tomorrow, or they are, at some point, but it will come up at some point in your career maybe. So then now we have basic definition of images, and let's talk about image processing a bit. So when you have an image already, then you already acquired it, so image acquisition already done. And then there are basic steps that you can follow. Usually it's not fitting perfectly, but it starts usually with some kind of pre-processing. So you pre-process the images to enhance them. And this organizes, this means also you organize your data, you name them correctly, but then also more operations directly on the image. For example, you enhance the contrast so you can actually see what structures you're interested in. You remove noise that might disturb the operations down the stream. Or if you have uneven background and other conditions, you can remove them. This is all part of general pre-processing of images. And then I have a few examples of that. For example, there's a contrast enhancement operation. I will give you over time a list that you can refer to of these standard operations. For example, where we can see here these structures better. I don't know what these are, but enhance or extraction of backgrounds because you are interested in it. Was done here to these cells, or the inverse operation where you, when you have the background, you can then of course remove it out of the image, just subtract it out, and this way you can get rid of this uneven background that will maybe disturb an algorithm down the line that expects an even background. Or noise removal, where you have all these small dots here that are noisy, that might get detected accidentally, and then you're going to remove them with this noise removal. Or data organization in general. If you have, for example, an image that has 5 channels, but you only need 3, then you remove the other ones. Or if you have images that have a different channel order, then you add operations that standardize them, so your workflow doesn't become an absolute mess later down the stream. And this is all basically pre-processing. Then you have something that's often called analysis. So very general term, and this means often something where you segment the structures that you're interested in, where you extract features that you're interested in, or for machine learning, which you can also do. You can apply that one or do some classification more. This is very general term, very general outline. And then some post-processing afterwards, where you collect the results, summarize them, add some filtering, generate your plots and visualizations, saving them out, etc. And it's a very general outline. It's taught everywhere, but in reality, it doesn't work. It's too general, and sometimes you have images that you maybe do some pre-processing, and then you do some analysis of some structure, but this allows to pre-process another part of the image in a way that you find other structures. So this can become more complex diagram in the end. And then sometimes you have post-processed data, and then, okay, I have this post-processing, this allows me to pre-filter something for the analysis goes back. And then this kind of general outline is often not so good. It's maybe a good start, but you will end up with analysis pipelines that usually don't look like that. They are looking very task-oriented to your data that you're working with. So let's then talk about tools for image enhancement a bit further. So I have here some examples again, where you can modify the images, where you can convert them or extract information. And then the idea is for you to have some toolbox that you can just go back through the slides here and then can refer to as where you can get started to do your own analysis. So, and for that we need to differentiate between different concepts and explain slowly how it's actually done on the pixel level and the algorithm level, so you understand what's going on there. So first of all, we have operations that are generally more global, so the whole image. For example, you tell an algorithm extract image statistics for the whole image. We say, and then calculates the minimum value or maximum value and the whole area, which is just width by height. But for this image, this is basically what image they calculated on that image. And then you can have the actually more interesting stuff, the local operations that apply some operation always in the vicinity of a pixel. And it's very, very useful. So how do they work is these local operations, they go through each pixel and then they define a local area around them. That, for example, on this pixel, it makes a small little window around it. And then it does something on it. And this something is then determines on what the operation actually will do. Usually, the most operations they will look at this pixel, calculate some value based on this surrounding pixel area, and then this way they calculate a new value and then store it at this location, but in a new image, because you don't want, when it goes here to the next pixel afterwards, to modify, to influence the calculations of the one before. And this is why it's always like taking two images. And I have a few concrete examples, so don't worry. And then later on, it will repeat that basically for other pixels, basically goes to the image like that. And this is basically the foundation of that. And for example, they are useful for noise removal. And noise has a lot of definitions, but the most common one for images, at least, is noise is just unwanted signal that you might not want. And two common noises are here this Gaussian noise, looks like that. I enhanced it with from the original image with some operations, so you see how it looks like. It follows just a Gaussian distribution is related to the sensor. And then also sometimes images have the salt and pepper noise, where you have these peaks or valleys, so holes or really high 1 pixel mountains. And this is caused in some way by the analog to digital conversion, at least. But although these operations are useful in general for many things. So how do we get rid of the noise? And one of the most basic operations is called the box filter, or mean filter is called different depending on the software. And the idea is that we are trying to estimate based on the surrounding value, because we are hopefully approximately in the same structure, the unnoised value from the surrounding values. So if we, for example, at a pixel like this is our pixel that we are currently want to work on, and this is our area around it. Then it looks like this value size, because we have here an 8-bit image. The center is 129, and then we have all these surrounding values like that. And then we are saying, okay, if we take the average pixel value of this area, this is maybe a good estimation of the actual true value of it. And then we calculated this average value. In this case, it's 128, so we correct one down and then write it into a new image. And then we repeat this basically for the whole image. So it's a very, very basic estimation. It also smoothes the image. You see it when you increase the window size. And it behaves differently for the noises. So when I increase this window size, so I can just also say it's a 5x5 window or even larger window around it that it uses for the estimation, then it will look different. So and you see that how it gets even more and more softened, right? Because at some point the window flows outside the structure that we are interested in, and it will just take in everything. For Gaussian noise, it actually works relatively well. You have to maybe cap it off at 3x3, and here it's already 2 noise. Although sometimes this is also useful if you are just generally interested in this cluster, and then if you just, as if you squint your eyes, just interested in a general structure, then some other algorithms might be able to find this one easier than the original one or one of those here. But for salt and pepper noise, this absolutely does not work. It makes these dots here even larger in the 3x3 window, and there are still artifacts left here in the 5x5. And then here it's anyway super destroyed regardless. So the box filter in this case is not perfect in performance as you probably want.
How does the box filter handle pixels at the edge of the image where the box would leave them?
Yeah, this is a good question because it's not defined. So when it's on the edge, imagine this is the edge and then we are going out of the image. Imagine this would be here the edge of it. Then the tool itself that implements it defines something. The true thing would be, so that if you mathematically correct, would be if you would stop here. So you can, with this 3x3 filter, you cannot go here because you would go outside. But then the more you apply it, then your image shrinks further. So this is no fun because at some point your image is gone and you lose information and the size changes. So the tools do different things. Sometimes they assume, okay, outside is 0, but then it will pull the average down on the elements on the side. Then, for example, this will be here darker because it sees, oh, this is outside, it's 0. Sometimes they don't calculate it in, then it will do something else. Well, it weighs the local pixels more. Sometimes they will mirror the image here, so they, when we have here pixel value 1, 2, 3, it will mirror it 1, 2, 3, like that, which has also its benefits and disadvantages. Sometimes they will make a period. There's no solution actually good, and it's really dependent on the image tool. This is where sometimes these differences come from between them, because here they are a bit weird how they behave. It's just like that. So something that you have to be aware of. These borders are sometimes anyway not good. So this is why. But for biological images, often if you have a cell, imagine you have a cell that goes here outside the border and then is cut off. Anyway, would it usually remove the data from your calculations? For example, you want to know the average cell size. Here, these are all in frame. Then I can calculate average size. But then if a cell is like half over the border, your brain infers, okay, it's maybe like that, but you don't know. So you rather remove it than ruin your statistics based on that. And then anyway, you have so much statistical power on a general microscopy image that you have enough data to remove something where you're really not sure. And there are then filters that handle it, but they are very tool specific. So yeah, box filter works not well for the salt and pepper noise, although it looks like an interesting effect. Then there are other filters, of course, because no one says you have to take the average of this local area. There's one other filter that's very popular, it's the median filter, and instead of an average, it takes the median pixel value. And then here in our case that I showed before, it would take the median value, and then it will be 130 because it's, if you sort it and then you take the center element, it's 130 this time. And this one behaves a bit differently when you run it on images. So the Gaussian noise, you see already, if you compare it, it's less blurry what it does. It keeps hard edges longer active, and then even here, where in the Gaussian filter, it's super blurry here. The edges are still there, although they are a bit expanded. And this is thus very useful for something where you want the edges to be preserved. And best case is actually here when we have salt and pepper noise. See, just even the 3x3 window, all your salt and pepper noise is basically gone, and the image is not even not so destroyed by this filter. So if you the smaller the filter, of course, the less assumption it makes on the surrounding pixel structure. And then we have only this singular pixel here left, but then if I increase the window to 5x5, then all salt and pepper noise is gone. And then I even set a very extreme case of salt and pepper noise, and still super easily gone with this filter. And then in the end, it almost looks the same as with the Gaussian noise. So this is a consideration. Sometimes you want specifically a box filter, and sometimes you want specifically a median filter, depending on how you want your image to behave in the following analysis steps. So that's why I show all of them, even if this box filter is a bit underperformed. And then what you can also do is, who says that you have to weigh all the elements in the local structure the same? So I have to come back again to the signal and noise estimation. The base idea, so let me repeat it, the base idea is that we are basically estimating the central value based on the surrounding images, under the hope that they are in the same structure. So we cover basically the value based on surrounding information. But this one here is more likely to, this pixel here is way more likely to be in the same structure, also this area around it, than something that is very far off it. Here you see already that black pixel is not anymore in this border wall here. So there are, and this is why filters were developed that basically account for that fact that the further away you are, that the less likely you are in the same structure. They should not count as much as one that is closer. So and here this is the general outset, like green is like I counted all the same. And then one very, very popular one, and you will find it in any tool that allows you to do custom image analysis, where you can pick the filters, is this Gaussian filter. And there the weight distribution is following a 2D Gaussian distribution. This is why it looks like that. The central element is like 0.69 or whatever that it counts of the whole thing, and then it decreases the further away you are from the center, and then this way you implement that. Also the box compared to the box filter. Also the box filter has a very mathematical reason why it's not good either, which I can explain if you're interested in that. Then it looks like that. If we have a weighted filter like that, I was very close, 0.68, not 0.69. So you have the image values that looks like that surrounding it. And then you have the so-called filter kernel. This is where it comes up, this kernel term that you will encounter everywhere. This filter kernel is basically just a weight matrix. So how much do pixels weigh? And then they calculate the new pixel value basically like this. You take your actual pixel value and then multiply it with the equivalent value in the same area in the filter area, and then sum it this times this plus this times this. I have it also as a slide. So this 15 times 0 plus 120 times 0 plus 140 times 0.1, and you do this for everything. The computer will do it fast for you because it's a computer. It will do this fast for you. Also, it has an optimization that can do this extremely fast by doing some transformations internally into a different space, into frequency space. And then this way it just calculates. And how does it behave? The Gaussian blur filter, the standard parameters you would use is the sigma and not anymore the size of the filter area. It's basically the sigma of the Gaussian, and then it adapts, it calculates the appropriate filter kernel size based on your sigma that you chose. And then behaves, it looks very different, very similar to the filter for box filter. Although you will at some point when you use it more see some minor differences. It still does the same destructive operation on the salt and pepper noise, but this time it looks round instead of square, and still super blurs everything. And it's actually very good at blurring things for you and mathematically correct, unlike the box filter. And then one additional thing. So generally is you would use median filter for something where you have edge preservation, and then instead of a box filter, you generally use the Gaussian filter rather than the Gaussian than the box filter, because it has better results. In box filter, you sometimes get a blockiness here in this area. Where do I do this? I don't see it here, but for larger images, sometimes you get in this box filter some weird blockiness. This comes from its mathematical roots being not really completely correct. Yeah. So, and then we have all these noise removal filters. Gaussian blur, box filter, although I know I just mentioned it, rather go with the Gaussian blur in general, and then the median filter, very good for edge preservation, even if your image, if you use a very large filter kernel, very useful. And then with these impulse noise or salt and pepper noise, performance varies depending on what you choose. Here you would rather go with the median filter to remove it. And this is why I explain so many different filters, because you have to choose which one works best and for your use case and what you intend to do with the image later on. Sometimes you explicitly, as mentioned before, you sometimes explicitly blur away structures because you are not interested in these individual cells here, but you want a cluster specifically. And then this way it can blur away any structures that might ruin your further analysis and just, okay, I care about the general area. So we have now image enhancement, but with noise removal, but there are other image enhancement tools, and specifically enhancement of contrast. And again, as with noise, there are different ways of defining contrast, and that you can look up online if you want. But one of them is this from Michelson. It's basically based on the min and max value, where the idea is that you ideally use the whole available value range of your image. So I made this image here super dark, and we obviously don't use its whole value range. We almost don't see it on the screen, because we have a value range from 8-bit here, 0 to 255, but we actually only use values from 8 to 63. And then this is why you cannot really see it. And based on that, you can come up with a very basic contrast enhancement, so-called histogram stretching. And it's literally that. You take the histogram or the pixel values and you stretch them so they take over the whole histogram space. So you make use of all the values that are available. There are different variants. You can sometimes say, okay, please keep the minimum at 8 and only expand the maximum to 255, or both. So it would go really from 0 to 255. Depends on your choices and what you need for the further pipeline downstream. But this is very simple and not always successful how you think it does. But there's a fancier histogram contrast enhancement, and it's called histogram equalization. It's something very different from stretching. Equalization, what it does, it runs some complicated mathematical algorithms that had to prove once in some image analysis class. I recommend to not try to prove it. It works. Where theoretically it will ensure that the histogram that comes out in the end is uniformly distributed. Obviously, it doesn't work because this is not uniformly distributed. But this would be basically the idea behind this is to maximize information. So it comes from all this information theory, this histogram equalization. It's a very complicated algorithm, doesn't work perfectly. As you can see, it's not a line because it assumes that you are working with real value pixel values, which you don't have. You have only 8-bit integral, and then it doesn't work like that. But it can enhance your image a lot if you use it correctly and really bring up structures. There are also in ImageJ especially additional options that you can control some additional parameters of it. You can say I want a specific saturation of the image to be achieved by histogram equalization and more. You can just explore them, try them out, how they behave, and then go with the operation that works best for you. That's anyway the general thing. And then I tried it here out in ImageJ or with JIPipe. This original image, you still see here how it ends at this 68. If I stretch just histogram, just stretch the values, then it looks like that. Then if I do histogram equalization that tries to make it uniform, it looks like that. I would rather go with this one here. So this doesn't mean that the fancy algorithm is always perfect and correct and you should always equalize it. There are cases where you want it to equalize. There are cases where you want it stretched, although in experience, usually stretching is absolutely fine, or whatever defaults ImageJ uses sometimes with its own contrast limits. Then we have some fancier things. We have sometimes images that have low contrast regions like this here. This is from some, I forgot the technology temporarily, how it's called. Electron microscopy. This is very low contrast, but this track here, I think, is very high contrast. And then these global contrast algorithms that just look at the histogram, they would see, oh, this image is super high contrast. I don't have to do so much because this is super dark and this is super bright. Good. Except maybe the equalization would do something. And fortunately, you can solve this, and there's a very, very useful algorithm, especially with for some microscopy images that have exactly that, where your contrast varies over the image. It's called contrast local adaptive histogram equalization. And ImageJ has it, and JIPipe has it too. And what it will do is it runs a histogram equalization, but locally. So it makes, again, a local window for each pixel, runs and then calculates how this pixel value should be adapted to fulfill the criteria. So it does a local contrast enhancement plus some contrast limiting. So CL contrast limited adaptive histogram equalization. It has some internal parameter to prevent it blowing up your image contrast where there is nothing. Otherwise, it would, for example, make this black part here super weird looking. And this works very, very well for many images. Just try it out as a slower algorithm, although not super slow, and can solve a lot of pain when you have unevenness in your image. And then you can run it through your pipeline. So usually maybe run it before you are doing your noise removal, and then later you can the noise removal tools will work better. And then basically from this toolbox that you have when you have an image, choose what is suitable for you. It's really up to the image. This is why I explain so many of these, even there are more, because then you have to really choose and just try out which one works best. And then we go to the next step, which is then thresholding. The thresholding is just basically turning your grayscale image that you get in, mostly it is grayscale image, sometimes it's a color image, into a binary image, where we then classify, is this background like this one here, or is this foreground like that one here? Theoretically, mathematics, you would save it in a 1-bit image because you need only yes or no, so 0 or 1. But computers cannot work with 1-bit natively, they need to work with 8 bits. So in ImageJ, this will end up being an 8-bit image. And then the general binarization rule depends also on the tool. Value 0 definitely is a background, and then everything larger than the value 0, being 0, is considered a foreground. Hopefully, when you have a binary image, it's usually clearly 255, because there are some tools who make the cutoff not at 0, but at 128. It depends a bit. So, but the thresholding algorithms usually make it 0 or 255 image for you. And you can, of course, set this manually. In ImageJ, it's very convenient where you can preview everything. In JIPipe, you can also do this, and then you type in the thresholds manually, the values specifically, and then you just come up with a range that works well for you. Then you can even say, this is everything below this value is discarded, is being seen as background, and then everything larger than this value here is seen as foreground. Or you can even set an upper limit, so if it's too bright, then it can be also seen as background. Or there's a bit of flexibility as well. And then find this usually in all image analysis tools for you, in even the commercial ones where you can then select it. But this is, of course, going back into your working manually, and which is what we want to discourage, because then there's a lot of work for you. For color thresholding, you can color, for example, with its native format, usually it's RGB images, and then you see quickly a limitation. The banana is obviously yellow for us humans. Do I see yellow here? No. I don't see yellow in here, and it makes it super hard to figure out what do I have to set here for my thresholds to pick out this yellow banana. And this is where these color spaces come back that I hinted at. That you can convert, depending on your image analysis tool, to different color spaces, where it's just a different representation of the same colors. Where you have then different settings or different thresholds that you can apply. The two common ones that are implemented generally in most image analysis software for scientific usages are HSB or HSV, usually the same. So hue, saturation, and brightness, where you have the hue, that is one of these base colors here. Then you saturate the color, either it's almost white, or like super desaturated, or super bright, or super high saturated, and then you set the brightness. It can represent the absolute same colors as RGB on your screen. And then the fancy one for scientific usages is more common. You don't usually find it in standard image, like image editing software like Photoshop. thilo. It's just a different way, where you have here luminosity, and then an axis between red and green, and then an axis between blue and yellow. It's just a different way. For example, JIPipe will help you convert them between these different types for usage. And then this way allows me to easily pick out the yellow through the manual thresholder. So I tried my best here, but you see here it's not perfect. It did not fully cover the banana, but the thilo space, I almost fell over that, covers here the banana way easier. You just have to play around with these manual thresholds. and it's sometimes valid for some data when they are very uniform and the auto threshold doesn't really work for you. But manual thresholding is really annoying, as said sometimes, because you have to sit there and try it out and fiddle around. And if you get new data that has different properties, then it suddenly doesn't work, and then you have to sit there again and fix it. So there are a lot of auto thresholding algorithms around that do the thresholding for you automatically based on different statistics that it can take from the image. And the most popular one that is everywhere shown, because it's very simple as well, is also thresholding. And then there are more, especially in ImageJ, it has like 16 different ones, isomodes or entropy-based ones. They all behave slightly different. For example, Ruman entropy is more lenient, and then takes also a bit darker pixels, while the also is more strict and wants more brighter pixels in general. So you have to try them out usually. That's why ImageJ has this try all threshold method. And also it's just so easy to explain. Always, I stole this here from Wikipedia, this explanation. So what it does is also expects basically that you have a bimodal distribution of your histograms. So all these thresholding methods that you see in ImageJ, they purely work on the histogram. I confirmed it because I looked in the code of ImageJ. Just takes the histogram and then calculates the threshold based on some statistics. And then also thresholding, what it does, it basically goes through the histogram of your image, goes through all available threshold values, and then it calculates for each single threshold the variance between classes. So you want basically the within the classes to be the image variance, the pixel variance, the minimal. Calculates like this interclass variance based on it, and then it just takes the maximum of this one. And it assumes the distribution, doesn't mean it does not work on images that don't follow this distribution. Just try it out, or try out the other thresholding methods. They are a bit data dependent on how well they work, and you get over time a feeling which one works for which case better. And then we can take this and combine it with local windows, because there are also local auto thresholding methods that then take a window and then applies for this singular pixel the threshold value that then decides if it's black or white. There are variants that you will find in ImageJ, local variants, like local also, local something that uses contrast, and then algorithms that are specifically built for local auto thresholding that have some limitations in there, so it does not accidentally detect super dark images. So imagine if your local window is in a spot that is particularly dark, so also would only see the histogram of this dark region and would do something, because it's an algorithm, it's dumb, it doesn't know anything. It will just do something and calculate a threshold based on what it sees. But then you have these special threshold, local thresholding methods here. These are the two that are implemented in ImageJ and in JIPipe as well, that do some additional checks to prevent your dark regions being exploded into, so your noise is getting exploded into wrong signal being thresholded. And I would try them out here if you have anything that has some, where the image is a bit varying in strength and you could not really correct it with other means. So we have thresholding now, but yeah, too short. I'm thinking about if I should do the pause now, because this is the next thickest one. Let's do the pause now, and then for like a quarter hour or something, or 10 minutes. So I'm back, and I talked to Sultan and our computer person Ahmed, and because I am faster than I think I was, than I want to be usually. So I asked Sultan, and if you want, he can talk about super resolution microscopy after me. Again, he will grab some slides that he has already made. So would be people interested in that? Okay, I assume yes. And then we can also do the computer handout session. So people who need laptops can then have laptops. After that, then we don't have to do it tomorrow. This is the plan. Because I'm again faster than I want to. Sometimes it happens. Okay, where we are at feature enhancement, and this is basically something that you would actually do after you do your enhancement and being your thresholding, and you will see why. So what is a feature? This is sometimes, when you look this up, different, but let's go with a definition that is most applicable for us. This generally a feature is an image region that you are interested in. It has some specific properties, and you want it, for example, to quantify it. And so far, we actually used a feature as well to find objects we are interested in. This was simple feature, it's just bright structures, like this. Bright pixels are the ones that we care about, and thresholding will well react to that one, because this is what they do. But this is often not enough. You sometimes want specific other things from the image that may or may not be bright, or have some other properties that differentiate it from other elements. For example, here's a small overview of certain general features that you will encounter anyways with images. You have like these ridges that you almost cannot see thanks to the beamer, so like these elongated structures, or you have blobs, which are just, they're called blobs, they are usually small circular structures, or you are not interested in the whole cell, maybe you're interested in the edges only. In the edge region, we have data also that where the edge is actually the interesting thing. Or here, images like that from transmitted light microscopy, where it's not, the structure is not bright, it's not generally brighter than the background, but it's like rougher. And these are the more general features. There might be more, depending on your use case, that you use or you want to specifically extract from your image. And then one of the very most common ones is something that has to do with edges. And fortunately, there are specific edge detectors that turn edges, the edges of some structures into bright pixels, so then our thresholding can pick them up. And there are different filters that you can use that you will find everywhere. So an ablation filter, a Sobel filter, and candy edge detector. They all behave differently. I feel examples on how they look. Which one you choose, you have to select yourself or try it out. And generally, if you take a profile of the image, like if I take a line profile here, and then this cell would look like that if it would be brighter, then the edge is basically the region where we have this, where the brightness increase or decrease is happening, so around here. And there are different operators, and I explain a bit how they work internally, and that's why I explained also these local concepts come all back. One popular one is the Laplacian edge detector, and what it is, it's basically just a local operation that uses a specific filter kernel. So the same principle that also does Gaussian blur and also does the median filter is applied here, rather Gaussian blur because you use a weighted matrix. And there are two variants of Laplacian, one that is looking also at corners, and then one that doesn't, it just looks at the north, south, east, west ones. And then there's something interesting about these filter kernels when they do edge detection. This is actually one of the main differentiators that you can quickly see when you look at the filter kernel. if they are an edge detector or if they are a smoothing filter, for example. If you sum the values inside an edge detector, they are 0. So there are 8 ones and then -8, 0. And here the same, 4 ones and then -4, 0. And then if you look at a Gaussian filter, then if you sum the values up, then there are 1, all of them in there. And this is really a differentiating factor. And this is exactly the same principle. So this is how they look, and this is what is produced. And this applies the same operation as before with filter kernels. And this is why ImageJ and the other tools, they have actually specific internal functions that basically take any of these filters, these filter kernels, and then apply it on the image. You can actually go on ImageJ and then JIPipe as well, and then provide a custom filter kernel if you want something extremely specific, and then apply it on the image fast, and then that's where you can customize your filters as well. Then there's another edge detector. This is the Sobel operator, and this actually has 2 different operations that are different. So first you have one that is a filter kernel that goes in a horizontal direction. Again, when you sum all these values, it's 0. And then you have one that goes in vertical direction, so X direction, Y direction. There's some formula missing. I don't know where it went. And then where is it? And then there's like somehow the formula got missing, a way to combine these together. I explained already now. Uncanny is, you see, it's a bit different. It makes, instead of a 8-bit image or whatever bit image here that is grayscale, it makes really a binary image with hard cutoffs, because the canny edge operation is actually a bit more complicated. It involves multiple operations in one that you unfortunately don't have to do. So the canny algorithm will do it for you. It will first apply an image smoothing to correct for some noise. And then my formulas are gone again. What it basically will do, it will calculate the Sobel in X direction and the Sobel in Y direction, both. And then the formula for gradient is missing. It basically combines the outputs of these 2 together to make an like a weighted average, or like an unweighted average always. So there's 2 different operations, one that uses a square root and one that just takes an average of it. The square root is a bit more precise. And then when it has this, looks like that here, then it will do some non-maximum suppression on the gradients, and then basically go along the edges. It finds the ones that are probably true edges, and then goes along the edges and figures out if a pixel, okay, is this a true edge? Yes, no, or maybe. And then basically this way really classifies in the end, is this an edge pixel? Yes, no. And this is why it makes such a hard cutoff and a thin line instead of something that is rougher. But you have to configure it correctly. There are additional parameters in there where you basically tell it where are the limits of that. What is the minimum value where you say this pixel or this gradient, this edge pixel is very likely a pixel, or maybe one, or definitely not. You can just try it out. In most image analysis tools have them, and then sometimes it's useful, sometimes it's not. Sometimes you rather like to go with the ablation and then do your auto-thresholding yourself, or with some additional smoothing or media filter, etc. There are different ways, sometimes only 1 or 2 works. Then we have another feature, it's also quite common in biological data. This is actually from kidney data. These are glomeruli. It's actually light sheet microscopy data that I received at some point. And what you can see, there are these blobs. So when you look from them from the side and the profile, it's basically a low variance round region that is quite bright. And there are specific operations that are really designed for pulling them up a bit, making them show a bit more. And there are a difference of Gaussian and gradation of Gaussian operations. And ImageJ, they don't have them, but JIPipe has them. So difference of Gaussian algorithm is basically applying two Gaussian filters. So it takes here the image, applies two Gaussian filters with different sizes on it, and then subtracting the images that come out of it. And this way, it really highlights this, to compare this one that is quite bright, but here we make it really, really bright. For the ones that are not so super highlighted, like this one here, it gets definitely brighter. We push it up compared to the background, and then this way, when we want the glomeruli, an auto-thresholding algorithm, when you set the correct one, maybe one that's a bit more lenient, like Huang, it will definitely find the most of the glomeruli that you are interested in. This one is the difference of Gaussian algorithm to do it for you. There's then also a variant of it, or opposite actually. The difference of Gaussian is an approximation of the true mathematical filter, Laplacian of Gaussian, which uses a weird G-shaped filter kernel, looks like that. When you look at the values, so negative values are red and green values are positive. When you look at it from the side, looks like this here, like a Mexican hat, how they sometimes call it. That's why it's sometimes called Mexican hat filter. And it looks very similar to what the difference of Gaussian does. It should, because the difference of Gaussian is an approximation of that one that is a bit faster, but it also pulls up really these blob features. And sometimes your image is a bit difficult, so you have to maybe go with the mathematical correct one and not the super fast one either. Then you have a feature that's called, I call ridges. Well, these here are, what was this? This is liver data, and these are the canaliculi, I think, from some liver data. Zoltan can explain it because I had data from him. And then you see here like these ridge-like features that were of interest because these needed to be measured, or something needed to be measured inside of them. And there are special operations for that you can actually use to find them or really enhance them. One is the morphological top hat filter, and then another are special vesselness filters that are really designed to handle such kind of data and really find vessels. They usually come from neuron analysis, these filters, because they want to trace the neurons, but they are useful for other things as well. They look from the profile view the same as blobs, because blobs are just round, and the ridges are basically if you draw out a blob like in MS Paint. So they look the same from the profile side. This is why you can actually use many ridge detector filters. Some of them at least you can use also to detect blobs. So for example, the top hat filter is one of those that you can see here from the liver data that it pushed down the background a lot and then really, really nicely highlights these canaliculi. And for this, and the same here for the glomeruli data, for the kidneys, it completely, almost completely got rid of the background, and they are super nicely shown now in the data. It's actually the filter that was used in the publication for that one. We used the top hat filter to extract the glomeruli in the light sheet data. This one especially works well, as we mentioned before, for both ridges and blobs, as seen here. And what it is a so-called morphological operation. I explained them in the later slide, I hope. That is basically a series of local minima and local maximum operations. It's just easier than the mathematical definition. What it does here is basically, okay, I keep my original image around, Is the image analysis tool will do it internally. Then on the image analysis tool, it will apply first a local minimum with a specific radius, and then so a specific area around it. And then after that, apply a local maximum as well with a specific radius. And then basically subtract the from the original this local maximum image. And then this way it generates this top hat image. Then there are vesselness filters that produce outputs like those. As mentioned before, they are made for usually by the people who do neuroscience to really trace the neurons in the microscopic images, but we can use them also for other vessel-like structures. And there are two that you will find usually in the standard libraries, the fungi vesselness and the ring vesselness. ImageJ specifically has the fungi vesselness, and this works very well. This is the one that I tried here. Ring vesselness has ImageJ doesn't have it, and it actually needs some input as well. And JIPipe's implementation also doesn't work perfectly. So you can try out in ImageJ-based environments, the fungi vesselness really works well, but you have to tune it a bit. You have to give it, for example, the approximate width of the vessel that you expect, then it can calibrate itself. And then something that you will encounter with transmitted light images. This is actually a set of filters that was developed by Sultan that he figured out. The problem was we have these transmitted light images and we really need these are macrophages. And as you know, you can of course stain them, but in this original data, the staining was not so successful and was really uneven and basically unusable. So the fluorescence data, if you remember, I showed it very shortly in the beginning. So this fluorescence data, there was a red channel, and there was supposed to be the macrophages, but it was not really working. So they only had the transmitted light images, and to really get the macrophages out. And it also, as additional things, you know that when you do staining, this influences the results. There's a paper, I think, here from one of the groups, I think it's even Bachhave's group, where was shown how much the staining actually influences the results. So if you have the option, you can go without staining. And Sultan developed this Hessian operator based filter that finds regions of low curvature. So when you look at the image, like a profile here, through one of these macrophages, it will look like that. This is our background, and then the macrophages, they have the same value, the same intensity approximately, but the curvature is different in there. And so you cannot apply just the thresholding because the threshold just looks at intensity. So what was developed is he used an algorithm that calculates a Hessian on the image. It's basically a local curvature measurement around each pixel. It's again one of these local filters. And then you see here that this curvature operator reacts very, very well towards where the cell is. And then, okay, we have they are merged together, but in this case it was not of concern. And then what he actually does, and I don't show it here, is you have this image, and there you can really use blurring. He applies a Gaussian blur to it, which then blurs together all these structures here because they are not of interest here for us. We just want a bright highlight against the background. So this is then all blurred away. And then you can just apply a thresholding, and then you get a quite good estimate of around the actual macrophages from that. And then there are special operations afterwards that can split it up, and I will list them hopefully. You can cut them up. Although today you just throw cell pros on that. Cell pros, so machine learning, deep learning will do this for you. It will find this basically, you give it an average size, and then it will find it for you. Although sometimes you have to train it, which JIPipe can do, and I don't know if Saturn will show it for you, but it's possible. So, and then you might have a binary image that comes out through thresholding that you achieve, but then it's not perfect often. Sometimes you have clusters, so stuff that merged and you rather want it apart. There's sometimes holes inside that you don't want this hole maybe because you want this cell filled out. For the cat image, we have here sometimes wrong segmentations, here, everywhere, disconnected parts. And this can be partially, not always, resolved by special operations that work either on the binary image or on a further step using regions of interest. These are extremely tool dependent. So ImageJ has a different tooling than, for example, Imaris for these. Why JIPipe usually has the ones from ImageJ because it's ImageJ based, but and they're also always differently named a bit. But general principles for binary images generally work. And there is one of those are these morphological operations. When you look them up here in these tutorials online, you find an image like that. Where you have an original image, and then you say you have a dilation operation. What it is actually just a local maximum. So it literally takes around each pixel a area, and then this pixel is the maximum value within this area. And then imagine if you are here, very at the border, and then your area covers the 1 pixel that is white here, then it will be colored red in the output, and this is how it's dilating. Then the opposite of it is erosion, where you take the local minimum in this surrounding area around each pixel. And this way you can thin the images down. These are the actually two basic operations, dilation and erosion, together with adding images or subtracting them, are all based around that. They form the basis of these morphological operations. Then you find also ones that are useful for example, getting rid of small objects that you don't want. The morphological opening operation would then get rid of these little objects here that were placed in there. What this is basically you just erode it. And as you've seen here before, erosion will basically shrink everything and then dilate it afterwards. It will increase it and inflate it again. So what then happens here is, first shrink it. This will leave my main structure alone, but these here are so small that shrinking them is making them delete. They're gone. And then the main object is then, of course, also shrink. So I inflate it back again to the original size. So basically do the inverse. But then still these small pixels here are gone. And then related operation to it, the morphological closing, which closes holes, which basically is just the opposite. I first dilate it, which then would basically inflate the whole object, but also inflate the object into these holes here. Then they are closed, and then when I erode it again with the same size, then the original object is restored, more or less. It's not perfect, as you can think. You see it even here, that this corner is not as before. So they are a bit disruptive, these morphological operations, and you should use them carefully where it's applicable. They tend to usually smoothen out angled features in the images. And then ones that are useful, here this morphological gradient. For example, if you want to make a quick outline, or you want just focus on the border of the image, so turn it to the border of the image. There are actually different gradient operations that are a bit different, like internal gradient and outer gradient, but they're all different variants of that. And then top hat, if you look at online, then this is absolutely not useful at all. But you have seen what it does. It basically suppresses the background around a bright structure, as with the glomeruli. And this is why when you look it up online, this definition of local minimum and local maximum is usually not the one that is given in resources. They rather give in textbooks a set-based definition, so mathematical sets, or something that is binary, but this falls apart for top hat. But you have seen how top hat works when you just see, okay, it's a local minimum and maximum. It will enhance these block-like and ridge-like structures in the images and suppress all backgrounds around it. So then we have also a few more binary operations as well that are extremely useful, that are not morphological operations technically, but sometimes they are called morphological operations because they are morphological, they change the shape. Differences between tools. For example, you have a hole filling algorithm, so you want to get rid of this hole here. Theoretically, you could just dilate it or morphologically close it, but then you would need to make the radius of this closing of this local area so large that it destroys the object. So there's no resemblance anymore of the original object. There you see really the destructiveness of these morphological operations, and you set them too high. The flat filling operator will solve this very nicely. They are filled and still the same as before. The outlines are not changed. So this is basically this flat filling algorithm. It's basically how your MS Paint flat fill works. It inverts first the image, then it starts a flat fill on the, usually it goes through the border and then takes everything that is white and then turns it, flat fills it to black. Looks like that here. And then you have basically the insides of these holes. You just add these images together. Once the holes not filled, then we have the fillings, boom, solved. But has a disadvantage because imagine if you have a structure inside a structure. So, for example, you have a cell that you can see the outline, but inside the cell there are objects that are of interest and they should be preserved. This will fill the whole cell. So choose the right tool for what you need. For this one, this flat fill is better. And then using these existing operations, you can make like a standard pattern, we would call it, where you can combine it together with these classical morphological operations. So this is literally from the kidney data and one of the published workflows that it does. This is a paper. It was a paper about a clearing method for light sheet fluorescence microscopy data, so doing the clearing of the sample that uses a clearing agent that is not toxic. And to showcase that it works properly, they had to, of course, measure the shrinkage of the tissue. This was done all in mouse kidneys. And for that, they, of course, had to estimate the volume. And then you can get the rough segmentation of the kidney itself very easily with thresholding. It is quite bright. I don't fortunately have here the original image, but you have it actually in your input data. And we will do it tomorrow as well in the example. It looks like that here, where when you just do Ruman threshold algorithm, but it has all these holes here inside. And then when you look at the border, even if you run the fill operation, the border looks super unclean and it's not correct and nice. So what can be then done to solve it is like this operation. This was also done in the application. They first dilate the whole kidney, so like you see it's inflated a bit. This will close up all these areas here that, because due to this dilation operations, will all be smoothened out. Then you fill the holes, the ones that are so large that the dilation will not cover them. And then it's basically the holes are filled, but the object is still too large. So you erode it, again, invert the dilation operation with the same size, and then it's back to the original size as it should be. But the holes are filled. And this is a pattern. We will implement it tomorrow together on this kidney data where we actually look at it in person. So see what will happen. And then clusters. They are annoying. Now they are mostly solved with self-host. But if you do it with classic image analysis, you need to usually, or sometimes need to separate clusters. So put cuts in here. It's also not perfect always. And then one method that basically is, okay, I can try it at first because it's so simple and you don't have to set anything up. It's called a distance transform watershed. What it does is basically a watershed transformation. So it's basically, good that I never explained what the watershed is. It basically starts at key points, and then basically they start flood filling from these points until they hit another area. Good that I don't have explanations for that one. But you can try it out in general. If it works, here it almost works, so it splits off some cells as I wanted, but these ones here are left alone. It's not perfect. So, but it's one good estimate for the start. And then this one here, we have to filter out with some additional object filtering usually to not disturb your statistics. Object filtering is extremely tool specific. ImageJ has its own tooling, Imaris has its own tooling, and Python also has tooling, but it calls it differently. And then, for example, you can use area or some brightness features based on some reference image or shape features, etc. There are so many options to filter out objects that you do not want. For example, if you know the size range of these cells here, these are fungal spores, you know the size. If this cluster here is way too large, way above the realistic size, then you can filter it out because it's probably a cluster that you do not want. Or measure for each object the average brightness of the image over that. And we will learn this specifically for JIPipe tomorrow, how JIPipe does it with this object filtering through its regions of interest system and Roy overlap system and so on. But this one will be all for tomorrow. So we now have a bit of a toolbox of different image analysis operations where you begin basically removing noise. Then you can do contrast enhancement of the image and push up the contrast of pixels you're interested in. Maybe you need to do some fancy operations like the contrast, the local histogram equalization. Then if your features are not already bright, like bright cells on a dark background, then you need to do some enhancements for the feature so they become bright compared to whatever you're not interested in. Then you can use different thresholding methods, for example, auto thresholding with Urzu, and then the resulting thresholded images you may or may not have to process further to achieve what you really want out of that. And then there are different tools that you will learn for JIPipe. And then actually I'm at the end of my part already. So I can maybe call on Soltan, then we can discuss with you further a bit about this super resolution microscopy. So I will be here and we will be back in 5 minutes or so.